we are learning about hormones. So first, proteins and peptide hormones are synthesized in the same fashion as other proteins. Steps in the synthesis of protein or peptide hormones is as follows. First, synthesized as large biologically inactive proteins. Prohormones cleaved to form smaller prohormones in the endoplasmic reticulum, then transferred to the Golgi apparatus for packaging into secretory vesicles. Enzymes in the vesicles cleave the prohormones to produce smaller biologically active hormones and inactive fragments. The vesicles are stored within the cytoplasm until their secretion is needed. And secretion of the hormones as well as the inactive fragments into the interstitial fluid or directly into the blood serum occurs by exocytosis. In many cases, the stimulus for exocytosis is increased cytosolic calcium concentration caused by the depolarization of the plasma membrane. In other instances, stimulation of an endocrine cell surface receptor causes increased cyclic adenosine monophosphate and subsequently activation of protein kinase that initiates secretion of the hormone. The peptide hormones are water soluble, allowing them to enter the circulatory system easily where they are carried to their target tissues. Steroid hormones are usually synthesized from cholesterol and are not stored. The chemical structure of steroid hormones is similar to that of cholesterol, and in most instances, hormones are synthesized from cholesterol. They are lipid soluble and are structurally related to cholesterol. And although there is usually very little hormone storage in steroid producing endocrine cells, large stores of cholesterol esters in cytoplasm vacuoles. So although there is usually very little hormone storage in steroid producing endocrine cells, large stores of cholesterol esters in cytoplasm vacuoles can be rapidly mobilized for steroid synthesis after a stimulus. Much of the cholesterol in steroid producing cells comes from the plasma, but there is also de novo synthesis of cholesterol in steroid producing cells. Because they are highly lipid soluble, they can simply diffuse across the cell membrane when synthesized and enter the interstitial fluid and then the blood. So, because they are highly lipid soluble, they can simply diffuse across the cell membrane when synthesized and enter the interstitial fluid and then the blood. Amine hormones are derived from tyrosine. The two groups of hormones derived from tyrosine are as follows: thyroid hormones and the adrenal medullary hormones. Adrenal medullary hormones in the sense catecholamines. Okay, catecholamines are also there. Which are the two types of amine hormones derived from tyrosine? The thyroid hormone and the adrenal medullary hormones or catecholamines. Okay. Then the thyroid hormones are synthesized and stored in the thyroid gland and incorporated into macromolecules of the protein thyroglobulin and is stored in large follicles until secretion. After entering the blood, most of the thyroid hormones combine with plasma proteins, especially thyroxine binding globulin, which slowly release the hormones to the target tissue. So, synthesis and secretion of peptide hormone. The stimulus for hormone secretion often involves changes in intracellular calcium or changes in cyclic adenosine monophosphate in the cell. Nucleus is there, DNA is there, transcription will be taking place, mRNA is there, synthesis will be taking place, translation, endoplasmic reticulum, Golgi apparatus, packaging will be taking place, then storage, secretory vesicles are there, extracellular fluid, into that exocytosis will be happening. So epinephrine and norepinephrine are formed in the adrenal medulla. Catecholamines are taken up into preformed vesicles and stored until secreted and released by exocytosis. Once the catecholamines enter the circulation, they can exist in the plasma in free form or in conjugation with other substances. Hormone secretion, transport and clearance from the blood. Hormone secretion after a stimulus and duration of action of different hormones. Hormone secretion after a stimulus and duration of action of different hormones. Some hormones such as norepinephrine and epinephrine are secreted within seconds after the gland is stimulated and may develop full action within another few seconds to minutes. The actions of other hormones such as thyroxine 2 growth hormone may require months for full effect. Thus, Each of the different hormones has its own characteristic onset and the duration of action, which each tailored to perform its specific control function. Then concentrations of hormones in the circulatory blood and hormonal secretion rates. The concentrations of hormones required to control most metabolic and endocrine functions are incredibly small. Their concentrations in the blood range from as small as 1 pg, which is 1 millionth of 1 millionth of a gram. 1 pg. Okay. Their concentrations in the blood range from as little as 1 pg, which is 1 millionth of 1 millionth of a gram, to each milliliter of blood up to at most a few micrograms, a few millionths of a gram per milliliter of blood. Similarly, the rates of secretion of various hormones are extremely small, usually measured in micrograms or milligrams per day. And transport of hormones in the blood. Water-soluble hormones, peptides and catecholamines are dissolved in the plasma. They are transported from their sites to synthesis to target tissues. So water-soluble hormones, peptides and catecholamines are dissolved in the plasma. They are transported from their sites of synthesis to target tissues. There they diffuse out of the capillaries into the interstitial fluid and ultimately to target cells. Water-soluble hormones, peptides and catecholamines are dissolved in the plasma. They are transported from their sites of synthesis to target tissues. There they diffuse out of the capillaries into the interstitial fluid and ultimately to target cells. Then steroid and thyroid hormones. These hormones, in contrast, circulate in the blood, mainly bound to plasma proteins. Usually less than 10% of steroid or thyroid hormones in the plasma exist free in solution. More than 99% of thyroxine in the blood is bound to plasma proteins. However, protein-bound hormones cannot easily diffuse across the capillaries and gain access to their target cells and are therefore biologically inactive until they dissociate from plasma proteins. Protein-bound hormones cannot easily diffuse across the capillaries and gain access to their target cells and are therefore biologically inactive until they dissociate from plasma proteins. The relatively large amount of hormones bound to proteins serve as reservoirs, replenishing the concentration of free hormones when they are bound to target receptors or lost from the circulation. Binding of hormones to plasma proteins greatly slows their clearance from the plasma. Clearance of hormones from the blood. Two factors can increase or decrease the concentration of a hormone in the blood: the rate of hormone secretion into the blood, the rate of removal of the hormone from blood. Metabolic clearance rate. So metabolic clearance rate is usually expressed in terms of the number of milliliters of plasma cleared of the hormone per minute. To calculate this clearance rate, one measures the rate of disappearance of the hormone from the plasma, nanograms per minute, and the plasma concentration of the hormone, nanograms per milliliter of plasma. And the metabolic clearance rate is calculated by the following formula. Metabolic clearance rate is equal to rate of disappearance of hormone from the plasma by concentration of hormone. Rate of disappearance of hormone from the plasma, concentration of hormone. Hormones are cleared from the plasma in several ways, including metabolic destruction by the tissue, binding with the tissue, excretion by the liver into bile, excretion by the kidneys into the urine. 4. Certain hormones, a decreased metabolic clearance rate may cause an excessively high concentration of the hormone in the circulating blood fluids. For example, when the liver is diseased, the steroid hormone levels increase because these hormones are conjugated mainly in the liver and then cleared into the bile. Most of the peptide hormones and catecholamines are water soluble and circulate freely in the blood. They are usually degraded by enzymes in the blood and tissues and rapidly excreted by the kidneys and liver, thus remaining in the blood for only a short period. For example, the half-life of angiotensin II circulating in the blood is less than a minute. And mechanism of action of hormones, hormone receptors and their activation. The first step of hormones activation is to bind to specific receptors on the target cell. Cells that lack receptors for the hormones do not respond. When the hormone combines with its receptor, this action usually initiates a cascade of reactions in the cell which with each stage becoming more powerfully activated so that even small concentration of the hormone can have a large effect. In human, hormone receptors are large proteins, and each cell that is to be stimulated usually has some 2,000 to 100,000 receptors. Also, each receptor is usually highly specific for a single hormone, which determines the type of hormone that will act on a particular tissue. So, the receptor is usually highly specific for a single hormone, which determines the type of hormone that will act on a particular tissue. Target tissue that are affected by a hormone are those that contain its specific receptors. The target tissue that are affected by a hormone are those that contain its specific receptors. Hormone receptors have been classified. Then hormone receptor interactions. This is a very important topic, so please pay attention. The number and sensitivity of hormone receptors are regulated. The number of receptors in a target cell usually does not remain constant from day to day or even from minute to minute. Receptor proteins are often inactivated or destroyed during the course of their function, and at other times they are reactivated or new ones are manufactured by the cell. For instance, increased hormone concentration and increased binding with its target cell receptors sometimes cause the number of active receptors to decrease. Downregulation of receptors. The decrease in the number of receptors in response to a high concentration of circulating hormones is referred to as downregulation. Downregulation of the receptors can occur as a result of inactivation of some of the receptor molecules, inactivation of some of the intracellular protein signaling molecules. Then classification of hormone receptors. The locations for the different types of hormone receptors are generally the following. In or on the surface of cell membrane, the membrane receptors are specific mostly for the protein, peptide, and catecholamine hormones. In the cell cytoplasm, the primary receptors for the different steroid hormones are found mainly in the cytoplasm. Next, in the cell nucleus, the receptors for the thyroid hormone is found in the nucleus and are believed to be located in direct association with one or more of the chromosomes, which are on the surface of the cell membrane, cell cytoplasm, cell nucleus. Then temporary sequestration of the receptor to the inside of the cell away from the site of action of hormones that interact with cell membrane receptors. Destruction of the receptors by lysosomes after they are internalized, or decreased production of the receptors. In each case, receptor downregulation decreases the target tissue's responsiveness to hormones. So, upregulation and downregulation of receptors. Downregulation, agonist will be there, antagonist will be there. Upregulation of receptors. An increase in the hormone receptor number as a consequence of a sustained low exposure to a particular hormone is called upregulation. Some hormones cause upregulation of receptors and intracellular signaling proteins. That is, the stimulating hormone induces greater than normal formation of receptors or intracellular signaling molecules by the target cell, or greater availability of the receptor for interaction with the hormone. When upregulation occurs, the target tissue becomes progressively more sensitive to the stimulating effects of the hormone. Then intracellular signaling after hormone receptor activation. Almost without exception, a hormone affects its target tissues by first forming a hormone receptor complex. Formation of this complex alters the function of the receptor, and the activated receptor initiates the hormonal effects. The various mechanisms of hormone receptor interactions are listed and explained as follows. Ion channel linked receptors, G protein linked hormone receptors, enzyme linked hormone receptors, intracellular hormone receptors. So, there are many different mechanisms or various mechanisms of hormone receptor interactions are listed and explained as follows. Ion channel linked receptors will be there. Ion channel linked receptors. Then G protein linked hormone receptors, enzyme linked hormone receptors, intracellular hormone receptors. First, we are gonna explain the ion channel linked receptors. Virtually all the neurotransmitter substances such as acetylcholine and norepinephrine combine with receptors in the postsynaptic membrane. This combination almost always causes a change in the structure of the receptor. Once again, I'm going to read ion channel linked receptor. Virtually all the neurotransmitter substances, acetylcholine and norepinephrine, combine with receptors on the postsynaptic membrane. This combination almost always causes a change in the structure of the receptor, usually opening or closing a channel for one or more ions. Some of these ion channel linked receptors open or close channels for sodium ions, others for potassium ions, other for calcium ions, and so forth. So first we are going to learn about ion channel linked receptors. We just mentioned them. Some of these ion channel linked receptors open or close channels for sodium ions, others for potassium ions, other for calcium ions, and so forth. So all the altered movement of these ions to the channels cause the subsequent effects on the postsynaptic cells. So remember, ion channel linked receptors, we will be having an example that is called neurotransmitters, just like that. So ion channel linked receptors and their mechanism. So next we are going to look into this picture. This is an ion channel linked receptors and their mechanism of action. Next, ion channel linked receptors and their mechanism. So here we could see an extracellular domain is there. Extracellular domain. Then cell membrane is there. Ion channel is there. Then transmembrane domain will be there. So in this depiction, we could see red colored neurotransmitter and blue colored ions. So neurotransmitter binds, ion channel opens, ions move through channel across cell membrane. Okay, this is as simple as that. And next, G protein linked hormone receptors. Next, G protein linked hormone receptors. Many hormone activated receptors that indirectly regulate the activity of target proteins. Enzymes or ion channels by coupling with groups of cell membrane proteins called heterotrimeric guanosine triphosphate binding proteins, G proteins, heterotrimeric guanosine triphosphate. Heterotrimeric guanosine triphosphate binding proteins or G proteins. These are over 1000 known G protein coupled receptors. All have 7 transmembrane segments that loop in and out of the cell membrane. Some parts of the receptor that protrude into the cell cytoplasm, especially the cytoplasmic tail of the receptor, are coupled to G proteins that include three in the sense trimeric. That's why we call them trimeric parts: the alpha, beta, and gamma subunits. And the ligand, which is a hormone, binds to the extracellular part of the receptor. A conformational change occurs in the receptor that activates the G protein and induces intracellular signaling that either open or close cell membrane ion channels or change the activity of an enzyme in the cytoplasm of the cell or activate gene transcription. So alpha, beta, and gamma subunits. And the ligand, which is hormone, bind to the extracellular part of the receptor. Conformational change occurs in the receptor that activate the G protein and induce intracellular signals that either open or close cell membrane ion channels or change the activity of an enzyme in the cytoplasm of the cell, activate the gene transcription. The intrinsic gene proteins are named for their ability to bind to guanosine nucleotides. In their inactive state, alpha, beta, and gamma subunits of G proteins form a complex that binds guanosine diphosphate on the alpha subunit. When the receptor is activated, it undergoes a conformational change that causes the GDP-bound trimeric G protein to associate with the cytoplasmic part of the receptor and to exchange GDP for GTP. Displacement of GDP by GTP causes the alpha subunit to dissociate from the trimeric complex and to associate with other intracellular signaling proteins. In turn, alter the activity of ion channels or intracellular enzymes such as adenylyl cyclase or phospholipase C, which alter cell function. The signaling event is terminated when the hormone is removed and the alpha subunit inactivates itself by converting its bound GTP to GDP. Then the alpha subunit once again combines with the beta and the gamma subunits to form an inactive membrane-bounded trimeric G protein. Some hormones are coupled to inhibitory G proteins denoted as GI proteins, where others are coupled to stimulatory G proteins denoted GS proteins. Inhibitory GI and GS stimulatory. This depending on the coupling of a hormone receptor to an inhibitory or stimulatory G protein. The hormone can either decrease or increase the activity of intracellular enzymes. Depending on the coupling of a hormone receptor to an inhibitory or stimulatory G protein, a hormone can either increase or decrease the activity of intracellular enzymes. Here we could see a picture which depicts the mechanism of action or activation of G protein coupled receptor. When the hormone activates the receptor, the inactivated alpha, beta and gamma G protein complex associate with the receptor and is activated with an exchange of guanosine triphosphate for guanosine diphosphate. This causes the alpha subunit to which the GDP is bound to dissociate from the alpha and beta subunits of the G protein and to interact with membrane-bound target protein or enzymes that initiate intracellular signals. Here we could see a receptor with G protein which is actually in the inactive form. GDP bound form. Alpha beta subunits are together in the trimeric form, and these proteins are called heterotrimeric guanosine triphosphate binding proteins. Heterotrimeric guanosine triphosphate binding proteins. And when hormone attaches into the receptor, you could see some changes happening. GDP changes into GTP that create the G protein to be active. Then alpha subunit detaches from beta and gamma and GTP activated target protein move into an effector in the sense that's another one. So GTP activated target protein, that is the enzyme. Okay. Then next, enzyme linked hormone receptors. Enzyme linked hormone receptors, some receptors when activated function directly as enzymes or are closely associated with enzymes that they activate. These enzyme linked receptors are proteins that pass through the membrane only once, in contrast to the 7 membrane G protein coupled receptors. Enzyme linked receptors have their hormone binding site on the outside of the cell membrane and their catalytic or enzyme binding site on the inside. When the hormone binds to the extracellular part of the receptor, an enzyme immediately inside the cell membrane is activated. When the hormone binds to the extracellular part of the receptor, the enzyme immediately inside the cell membrane is activated or occasionally inactivated. When inhibition is taking place, there will be an activation. And although many enzyme-linked receptors have intrinsic enzyme activity, others rely on enzymes that are closely associated with the receptor to produce changes in cell function. Cytokines are some peptide growth factors and hormones that use enzyme-linked receptor tyrosine kinase for cell signaling. One example of an enzyme-linked receptor is the leptin receptor. Leptin is a hormone secreted by fat cells and has many physiological effects, but it is especially important in regulating appetite and energy balance. The leptin receptor is a member of a large family of cytokine receptors that do not themselves contain enzymatic activity but signal through associated enzymes. In this case of leptin receptor, one of the signaling pathways occur through a tyrosine kinase of the Janus kinase family, JAK2. Some of the effects of leptin occur rapidly as a result of activation of these intracellular enzymes, whereas other actions occur more slowly and require synthesis of true new proteins. Another example, and widely used in hormonal control of cell function, is for the hormone to bind with a special transmembrane receptor, which then become the activated enzyme adenylyl cyclase at the end that protrudes into the interior of the cell. This cyclase catalyzes the formation of CAMP, which has a multitude of effects inside the cell to control cell activity. CAMP is called a second messenger because it is not the hormone itself that directly institutes the intracellular changes. Instead, the CAMP serves as a second messenger to cause these effects. For a few peptide hormones such as atrial natriuretic peptide, cyclic guanosine monophosphate, which is only slightly different from CAMP, serves in a similar manner as a second messenger. In case of an enzyme-linked receptor, for example, the leptin receptor, the receptor exists as a homodimer, two identical parts. The leptin binds to the extracellular part of the receptor, causing phosphorylation and activation of the intracellular associated Janus kinase 2, JAK2. This causes phosphorylation of signal transducer and activator of transcription STAT proteins, which then activates the transcription of target genes and the synthesis of proteins. JAK2 phosphorylation also activates several other enzyme systems that mediate some of the more rapid effects of leptin, specific tyrosine phosphorylation sites. Leptin is there, leptin receptor, JAK2, STAT3, activation of enzymes, physiological effects, translation, and mRNA and target gene is also produced. Hormones that use receptor tyrosine kinase signaling: fibroblast growth factor, growth hormone, hepatocyte growth factor, insulin-like growth factor 1, leptin, prolactin, vascular endothelial growth factor. Then, for a few polypeptide hormones such as atrial natriuretic peptide, cyclic guanosine monophosphate (cGMP), which is only slightly different from CAMP, serves in a similar manner as a second messenger. Then intracellular hormone receptors and activation of genes. Several hormones including adrenal and gonadal steroid hormone, thyroid hormones, retinoid hormones, and vitamin D bind with the protein receptors inside the cell rather than in the cell membrane. Before that, cyclic adenosine monophosphate mechanism by which many hormones exert their control of cell function. Adenosine diphosphate ADP, adenosine triphosphate ATP. Extracellular fluid is there, cytoplasm is there, hormone is there. GTP binds to adenylyl cyclase, then ATP, CAMP, then activate CAMP dependent protein kinase, inactivate CAMP dependent protein kinase, lead to protein ATP, ADP plus protein PO4. Then intracellular hormone receptors and activation of genes. Several hormones including adrenal and gonadal steroid hormones, thyroid hormones, retinoid hormones, and vitamin D bind with the protein receptors inside the cell rather than in the cell membrane. Because these hormones are lipid soluble, they readily cross the cell membrane and interact with the receptors in the cytoplasm or nucleus. The activated hormone receptor complex then binds with a specific regulatory promoter sequence of the DNA called the hormone response element, and in this manner either activates or repress transcription of specific genes and formation of messenger RNA. So don't forget this word, hormone response element. Hormone response element. Hormone response element. Therefore, minutes, hours, or hormone response element, and in this manner, the activated hormone receptor complex then binds with a specific regulatory sequence of the DNA called the hormone response element, and in this manner either activates or represses transcription of specific genes and formation of messenger RNA. Therefore, minutes, hours, or even days after the hormone has entered the cell, newly formed proteins appear in the cell and become the controllers of new or altered cellular functions. Many different tissues have identical intracellular hormone receptors, but the genes that the receptors regulate are different in the different tissues. An intracellular receptor, an intracellular receptor can activate the gene response only if the appropriate combination of gene regulatory proteins is present, and many of these regulatory proteins are tissue specific. Thus, the response of different tissues to a hormone are determined not only by the specificity of the receptor but also by the expression of genes that the receptors regulates. Then, genomic and non-genomic effects of hormones. The actions of several hormones listed previously, which act on receptors and within a cell or nucleus, causing gene transcription, target gene expression, and ultimately cellular response, are referred to as genomic effects of a hormone. More recently, novel methods of signal transduction through steroid hormone receptors that do not involve gene transcription have been identified for several hormones. So, more recently, novel methods of signal transduction through steroid hormone receptors that do not involve gene transcription have been identified for several hormones. These non-genomic effects do not depend on gene transcription or protein synthesis. Novel methods of signal transduction through steroid hormone receptors that do not involve gene transcription have been identified for several hormones. These non-genomic effects do not depend on gene transcription or protein synthesis. This novel mechanism involves steroid-induced modulation of cytoplasmic or cell membrane-bound regulatory proteins. The hormones that have both genomic and non-genomic effects include estrogens, progestin, aldosterone, and androgens. The characteristics of non-genomic effects include the following. The actions are usually too rapid, seconds to minutes, to involve any changes in mRNA and protein synthesis. The actions are usually too rapid, seconds to minutes, to involve any changes in mRNA and protein synthesis. These actions can be observed even in highly specialized cells that do not accomplish mRNA and protein synthesis. For example, spermatozoa. The non-genomic effects can be elicited even by steroids coupled with high molecular weight substances that do not pass across the plasma membrane and do not enter into the cell. Non-genomic effects are not blocked by inhibitors of mRNA or protein synthesis. Non-genomic effects are not blocked by antagonists of the classic genomic steroid receptors. When we try to learn about mechanism of interaction of lipophilic hormones such as steroids with intracellular receptors in target cells, after the hormone binds to the receptor in the cytoplasm or on the nucleus, the hormone receptor complex binds to the hormone response element, which is acting as a promoter on the DNA. This either activates or inhibits gene transcription, formation of messenger RNA or mRNA, and protein synthesis. When we try to closely observe this picture, you could see lipophilic hormone directly diffuse into the cytoplasmic side. Then cytoplasmic receptor is there. Then hormone receptor complex is formed. Nuclear receptor is there. No, that's another mechanism. Then hormone receptor complex is formed. Then hormone response element is activated in DNA. Later mRNA is formed, which produce protein. Target cell is there, proteins are produced, ribosome is there, mRNA, nuclear envelope, and nuclear 4 is there. This is a very important mechanism. Mechanism of interaction of lipophilic hormones such as steroids with intracellular receptors and target cells. First mechanism is cytoplasmic receptor. Next one is receptors on the nucleus. So nuclear receptors are also there. Okay. The next major topic is second messenger mechanisms for mediating intracellular hormonal functions. This is going to be a bit complex, so I will be slowly reading this along with showing some important pictures. So let's start. Second messengers are molecules or substances that help communicate signals from the cell surface to specific target sites within the cell, either in the cytoplasm or nucleus. Once again, second messengers are molecules or substances that help communicate signals from the cell surface to specific target sites within the cell, either in the cytoplasm or nucleus. Second messengers help in significantly amplify the strength of the signals. There are several types of second messengers. We noticed earlier that none of the means by which hormones exert intracellular actions is to stimulate formation of the second messenger CAMP inside the cell membrane. The CAMP then causes subsequent intracellular effects of the hormone. Thus, the only direct effect that the hormone has on the cell is to activate a single type of membrane receptor. The second messenger does the rest. So CAMP is not the only second messenger used by the different hormones. Two others, especially important ones, are calcium ions and associated calmodulin, products of membrane phospholipid breakdown. Adenylyl cyclase CAMP second messenger system. Binding of the hormone with the receptor allows coupling of the receptor to the G protein. If G protein stimulates the adenylyl cyclase CAMP system, this is called the GS protein. Adenylyl cyclase CAMP system, denoting a stimulatory G protein. Stimulation of adenylyl cyclase, a membrane-bound enzyme, by the GS protein, then catalyzes the conversion of a small amount of cytoplasmic adenosine triphosphate into CAMP inside the cell. So, stimulation of adenylyl cyclase, a membrane-bound enzyme, by the GS protein, then catalyzes the conversion of a small amount of cytoplasmic adenosine triphosphate into CAMP inside the cell. This then activates CAMP-dependent protein kinase, which phosphorylates specific cell proteins, triggering biochemical reactions that ultimately lead to the cell's response to the hormone. Then, adenosine triphosphate into CAMP inside the cell. This then activates CAMP-dependent protein kinase, which phosphorylates specific cell proteins, triggering biochemical reactions that ultimately lead to the cell's response to the hormone. Once CAMP is formed inside the cell, it usually activates a cascade of enzymes. In this way, even the slightest amount of hormone acting on the cell surface can initiate a powerful cascading activity force for the entire cell. If binding of the hormone to its receptor is coupled to an inhibitory G protein denoted by GI, adenylyl cyclase will be inhibited, reducing formation of CAMP and ultimately leading to an inhibitory action in the cell. Then next, cell membrane phospholipid second messenger system. Some hormones activate transmembrane receptors that activate the enzyme phospholipase C attached to the inside projections of the receptor. So you are learning about cell membrane phospholipid second messenger system. That was the next one. Some hormones activate transmembrane receptors that activate the enzyme phospholipase C attached to the inside projections of the receptors. This enzyme catalyzes the breakdown of some phospholipids in the cell membrane, especially phosphatidylinositol biphosphate (PAP2), into two different second messenger products, which are the IP3 and DAG, inositol triphosphate and diacylglycerol. Okay. Some hormones activate transmembrane receptors that activate the enzyme phospholipase C attached to the inside projections of the receptor. These enzymes catalyze the breakdown of some phospholipids in the cell membrane, especially phosphatidylinositol biphosphate (PAP2), into two different second messenger products, inositol triphosphate, diacylglycerol (DAG). So the IP3 mobilizes calcium ions from mitochondria and endoplasmic reticulum, and the calcium ions then have their own second messenger effects such as smooth muscle contraction and changes in cell secretion. DAG, on the other lipid second messenger, activates the enzyme protein kinase C, which then phosphorylates a large number of proteins leading to cell's response. In addition to these effects, the lipid portion of the DAG is arachidonic acid, which is the precursor of the prostaglandins and other local hormones that cause multiple effects in tissues throughout the body. I'm going to break down this topic into small chunks once again. We are learning about cell membrane phospholipid second messenger system. Some hormones activate transmembrane receptors that activate the enzyme phospholipase C attached to the inside projections of the receptors. These enzymes catalyzes the breakdown of some phospholipids in the cell membrane, especially phosphatidylinositol, into two different second messenger products, inositol triphosphate IP3 and diacylglycerol DAG. So what this IP3 does is that the IP3 mobilizes calcium ions from mitochondria and the endoplasmic reticulum, and the calcium ions then have their own second messenger effects such as smooth muscle contraction and changes in cell secretion. DAG, on the other lipid second messenger, activates the enzyme protein kinase C, which then phosphorylates a large number of proteins leading to the cell's response. In addition to this effect, the lipid portion of DAG is arachidonic acid, which is a precursor for the prostaglandins and other local hormones that cause multiple effects in tissues throughout the body. Then third system of second messenger was calcium calmodulin second messenger system. Third method is calcium calmodulin second messenger system. Another second messenger system operates in response to the entry of calcium ions into the cells. Calcium entry may be initiated by changes in membrane potential that open calcium channels, or a hormone interacting with the membrane receptor that open calcium channels. Upon entering a cell, calcium ions bind with the protein calmodulin. The calmodulin changes its shape and initiates multiple effects inside the cell, including activation or inhibition of protein kinase. Activation of calmodulin-dependent protein kinase causes, via phosphorylation, activation or inhibition of proteins involved in the cell's response to the hormone. For example, one specific function of calmodulin is to activate myosin light chain kinase, which acts directly on the myosin of smooth muscle to cause smooth muscle contraction. The normal calcium ion concentration in most cells of the body is 10 raised to minus 8 or 10 raised to minus 7 mole by liter, which is not enough to activate the calmodulin system. But when the calcium ion concentration raises from this normal value to 10 raised to minus 6 to 10 raised to minus 5 mole per liter, enough binding occurs to cause all the intracellular actions of calmodulin. Okay. So in this picture, we are going to learn the previously mentioned type, the cell membrane phospholipid second messenger system, by which some hormones exert their control of cell function. Diacylglycerol, inositol triphosphate, phosphatidylate inositol triphosphate. Extracellular fluid is there, cell membrane is there, peptide hormone binds to the receptor, G protein is there, phospholipase C is activated, DAG plus IP3, PIP2 will be turning into DAG plus IP3. Then cytoplasm is there, active protein kinase C is there, inactive protein kinase C got activated. Then PO4 protein endoplasmic reticulum CA2 plus cells response will be something related with the calcium release. And the next, calcium calmodulin second messenger system. Here we could see a picture which depicts this. Extracellular fluid is there, cell membrane is there, first messenger binds with receptor, intracellular fluid is there, increased calcium entry via cell membrane calcium channels, increased calcium is released from endoplasmic reticulum. Increased calcium in the cytosol, inactive calmodulin lead to active calmodulin, active calcium calmodulin, then inactive calmodulin dependent protein kinase, active calmodulin dependent protein kinase, then protein PO4 ADP cellular response is there. Okay. Then next, hormones that act mainly on the genetic machinery of the cell. Then hormones that act mainly on the genetic machinery of the cell. Steroid hormone increase protein synthesis. Another means by which hormones act, especially the steroid hormones, is to cause synthesis of proteins in the target cells. These proteins then function as enzymes, transport proteins or structural proteins, which in turn provide other functions of the cell. The sequence of events in steroid function is essentially the following. The steroid hormone diffuses across the cell membrane and enters the cytoplasm of the cell, where it binds with a specific receptor protein. The combined receptor protein hormone then diffuses into or is transported into the nucleus. The combination binds at specific points in the DNA strands in the chromosome, which activates the transcription process of specific genes to form mRNA. Once again, the steroid hormone diffuses across the cell membrane and enters the cytoplasm of the cell, where it binds with a specific receptor protein. The combined receptor protein hormone then diffuses into or is transported into the nucleus. The combination binds at specific proteins on the DNA strands in the chromosome. Wait a minute, I find it a bit difficult to learn, so I'm gonna read this once again. Sorry. The sequence of events in steroid function is essentially the following. The steroid hormone diffuses across the cell membrane and enters the cytoplasm of the cell, where it binds with a specific receptor protein. Yeah. The combined receptor protein hormone then diffuses into or is transported into the nucleus. Yeah, that's right. This combination binds at specific points on the DNA strands in the chromosome, which activates the transcription process of specific genes to form mRNA. The mRNA diffuses into the cytoplasm, where it promotes the translation process at the ribosomes to form new proteins. To give an example, aldosterone, one of the hormones secreted by the adrenal cortex, enters the cytoplasm of renal tubular cells, which contain a specific receptor protein often called the mineralocorticoid receptor. Mineralocorticoid receptor. After about 45 minutes, proteins begin to appear in the renal tubular cells and promote sodium reabsorption from the tubules and potassium secretion into the tubules. Thus, the full action of the steroid hormone is characteristically delayed for at least 45 minutes up to several hours or even days. This action is in marked contrast to the rapid actions of some of the peptides and amino acid derived hormones such as vasopressin and norepinephrine. Then thyroid hormone increase gene transcription in the cell nucleus. The thyroid hormone tyrosine and triiodothyronine cause increased transcription by specific genes in the nucleus. To accomplish this increased transcription, these hormones must bind directly with the receptor protein in the nucleus, These receptors are activated transcription factors located within the chromosome complex, and they control the function of the gene promoters. The following two features of thyroid hormone function in the nucleus are important. They activate the genetic mechanism for the formation of many types of intracellular proteins, probably 100 or more. Many of these intracellular proteins are enzymes that promote enhanced intracellular metabolic activity in virtually all cells of the body. Once bound to the intranuclear proteins receptors, the thyroid hormone can continue to express their control functions for days or even weeks. Then measurement of hormone concentrations in the blood. Till now, we learned some important aspects related to hormones and their receptors. Now we are going to learn something related to a practical application. Practical application in the sense something related to laboratory application or something like that. So let's learn about that. Most hormones are present in the blood in extremely minute quantities. Some concentrations are as low as 1 billionth of a milligram per milliliter. Therefore, it was difficult to measure these concentrations by the actual chemical means. However, an extremely sensitive method that was developed about 50 years ago revolutionized the measurement of hormones, their precursors, and their metabolic end products. This method is called radioimmunoassay. More recently, additional methods such as enzyme-linked immunosorbent assay (ELISA) tests have been developed for accurate, high-throughput measurements of hormones. So let's quickly learn about both these techniques as we have got less time. Okay, so radioimmunoassay. The method of performing radioimmunoassay is as follows. First, an antibody that is highly specific for the hormone to be measured is produced. Second, a small quantity of this antibody mixed with a quantity of fluid from the animal containing the hormone to be measured, mixed simultaneously with an appropriate amount of purified standard hormone that has been tagged with a radioactive isotope. However, one specific condition must be met. There must be too little antibody to bind completely both the radioactively tagged hormone and the hormone in the fluid to be assayed. Therefore, the natural hormone in the assay fluid and the radioactive standard hormone compete for the binding sites of the antibody. In the presence of competing, the quantity of each of these two hormones, the natural and the radioactive that binds, is proportional to its concentration in the assay fluid. Third, after binding has reached equilibrium, the antibody hormone complex is separated from the remainder of the solution, and the quantity of radioactive hormone bound to this complex is measured by radioactive counting technique. If a large amount of radioactive hormone has bound with the antibody, it is clear that there was only a small amount of natural hormone to compete with radioactive hormone, and therefore the concentration of natural hormone in the assayed fluid was small, and the converse is also true. So fourth, to make the assay highly quantitative, the radioimmunoassay procedure is also performed for standard solutions of untagged hormone at several concentration levels. Then a standard curve is plotted. By comparing the radioactive counts recorded from the unknown assay procedures with the standard curve, one can determine within an error of 10 to 15% the concentration of hormone in the unknown assayed fluid. As little as a billionth or even trillionth of a gram of hormones can often be assayed in this method. The next, enzyme linked immunosorbent assay. ELISA can be used to measure almost any protein, including hormones. The test combines the specificity of antibodies with the sensitivity of simple enzyme assays. Basic elements of this method, which is often performed on plastic plates that can have 96 small wells. Each well is coated with an antibody, AB1, which is specific for the hormone being assayed. Samples of standards are added to each of these wells, followed by a second antibody, AB2, that is also specific for the hormone but binds to a different site of the hormone molecule. A third antibody, AB3, which is added, recognizes AB2 and is coupled to an enzyme that converts a suitable substrate to a product that can be easily detected by colorimetric or fluorescent optical methods. Because each molecule of enzyme catalyzes the formation of many thousands of product molecules, even small amount of hormone molecules can be detected. In contrast to competitive radioimmunoassay methods, ELISA methods use excess antibodies so that all hormone molecules are captured in antibody-hormone complexes. Therefore, the amount of hormone present in the sample or in the standard is proportional to the amount of product formed. The ELISA method has become widely used in clinical and research laboratories because it does not use radioactive isotopes. Much of the assay can be automated using 96 well plates, and it has proved to be a cost-effective and accurate method for assessing hormone levels.